Press record. Awesome. Thank you so much. So welcome everyone, and thank you for joining PARC's Disease Task Team webinar, History and Current Knowledge of Snake Fungal Disease. My name is Jenna Palmisano. I am the chair of the National PARC Disease Task Team and a PhD student in Dr. Anna Savage's lab at the University of Central Florida. I study the genomic and physiologic impacts of emerging pathogens on pygmy rattlesnakes, including fungal pathogens that Jeff and Ellen will be talking about today. So the mission of the PARC Disease Task Team is to facilitate and guide communication and collaboration on herpetofaunal diseases among PARC regions, federal and state agencies, and partners. Our goal with the webinar series is to help connect science and management action by providing information to land and natural resource managers. And so we welcome any input and feedback that you may have. So before I introduce our speakers today, a little housekeeping. Captioning is available by clicking the CC button at the bottom of your screen. And this webinar is recorded and will be posted on the PARC Disease Task Team webpage and YouTube. And for now, all attendees will be muted until the Q&A session at the end, but you can ask questions in the chat during the presentation. We have a few members and leaders of the PARC Disease Task Team that will be monitoring the chat for questions, and they might be able to answer within the chat feature, or will collect the other questions for the Q&A session at the end. So now to introduce our speakers, Dr. Jeff Lorch is a microbiologist at the National Wildlife Health Center, where he specializes in disease ecology and the management of emerging wildlife diseases. His research focuses on bat white nose syndrome, snake fungal disease, and the surveillance of bee sow. Dr. Lorch also develops molecular methods for detecting wildlife pathogens and holds a PhD in molecular and environmental toxicology from the University of Wisconsin-Madison, where he also earned bachelor's degrees in bacteriology and wildlife ecology. Since joining the USGS in 2015, Dr. Lorch has focused his career on investigating wildlife mortality events and advancing pathogen detection methods. He is also an honorary associate in pathobiological sciences at the University of Wisconsin-Madison. Dr. Ellen Haynes is an assistant research scientist at the Southeastern Cooperative Wildlife Disease Study at the University of Georgia, and the Regional Wildlife Health Coordinator for the Southeastern Association of Fish and Wildlife Agencies. Ellen provides wildlife disease consultation and research coordination for state agencies, and she has a special interest in the epidemiology of infectious diseases and free-ranging North American wildlife, with a particular focus on amphibians and reptiles, especially snakes. She earned her DVM from Cornell University and her PhD in ecology, evolution, and conservation biology from the University of Illinois, Urbana-Champaign, where she conducted extensive work on snake fungal disease. In addition to her research, Dr. Haynes is the co-chair of the PARC Southeastern Regional Disease Task Team. So on behalf of PARC, we'd like to thank PARC's Disease Task Team for helping to develop this webinar with the speakers in a variety of products that will be mentioned in this talk, as well as disease-specific fact sheets available on our website, and we can resend that link. And now I'm pleased to welcome our speakers for today's webinar, Dr. Jeff Lorch and Dr. Ellen Haynes. Jeff and Ellen, take it away.
Good afternoon, everybody, and thank you, Jenna, for the introduction, and thank you for the PARC Disease Task Team for inviting us to speak today. We are excited to be here and talk about snake fungal disease with you all. To start, so here's an outline of the topics we'll be covering today. First, history of the disease and the geographic and species distributions, then moving into additional pathogens associated with this disease, the pathogenesis, testing and diagnosis, pathogen origins and epidemiology. And then finally, we'll cover conservation implications, management and treatment options, decontamination and best field practices, and then cover some ongoing research and future directions where more research is needed. Jeff and I will be tag teaming these topics, so we'll be bouncing back and forth, and then there'll be a question and answer session at the end. So before we delve into the specifics of this disease, I always like to point out that disease is just one threat to wildlife health and Earth's biodiversity. Human activities have had massive impacts on the global landscape, including deforestation and climate change. And these activities result in biodiversity loss through a variety of mechanisms, including habitat destruction and increasing the transfer of pathogens between wildlife, domestic animals, and people. So studying wildlife diseases is really crucial to protecting global health and biodiversity. Emerging fungal pathogens are of particular concern because of their high virulence or ability to cause illness, long-lived environmental stages, and ability to infect a wide range of hosts as opportunistic pathogens. Fungal diseases in wildlife that you may already be familiar with include white nose syndrome in bats, Ophidiomyces, which affects amphibians, Epidemiology, which causes shell disease in aquatic turtles, and of course, our topic for today, snake fungal disease.
Right, so we thought we'd start by presenting a little bit of history about snake fungal disease, and here we're sort of focusing, and throughout this talk, we'll probably mostly be focusing on Ophidiomyces or Ophidiomyces, unless we state otherwise. So looking back in reports in the literature, we see evidence of infections that we suspect were snake fungal disease or Ophidiomyces going back into at least the 1960s. Often these were reported under terms like hibernation blisters or hibernation sores. There always wasn't much of an investigation to determine what the causative agent was. And so these are sort of hypothetical cases. Most of them seem to be pretty mild. You can kind of see in that upper photo there of a snake that has a mild lesion comparable to what we often see mentioned in the earlier literature. And then in 1986, we have the first confirmed case of Ophidiomyces. And this is sort of diagnosed retrospectively. So 1986, a captive snake in New York was known to have a fungal infection, and that fungal isolate was saved. And upon going back and characterizing that fungal isolate in more modern times, we're able to identify it as Ophidiomyces. But again, that's in a captive animal in North America. The 1990s, there was a study done in Florida where there was an outbreak of skin disease in pygmy rattlesnakes. A fungal etiology was identified, but it was probably misidentified as to species. And going back and resampling those same populations in the 2000s suggested that those probably were indeed cases of Ophidiomyces. And in 2006, similarly, we have sort of these unconfirmed instances in rattlesnakes in a population in New Hampshire, where there was a pretty significant decline in that population in association with severe skin infections. Wasn't confirmed to be Ophidiomyces, but based on the case histories and in clinical signs, we feel pretty comfortable in saying that it most likely was caused by Ophidiomyces. And then 2009 is actually when Ophidiomyces is recognized as a species of fungus. This came from a black rat snake. You can see the photo there in the center. It was a captive animal, but it had been wild caught in Georgia. And then shortly thereafter, in 2010, is when snake fungal disease really starts to gain attention. That's when these pretty severe infections in eastern Massasauga rattlesnakes that you can see there in the lower corner are reported in Illinois. But even predating some of these reports in the literature, by kind of going back and looking in museum specimens, we're able to determine that Ophidiomyces has actually been present in wild snakes in North America for even longer. And so these are confirmed cases that we were able to look at microscopically and detect the pathogen in some of these museum specimens that have been preserved properly. And our earliest detection in those cases is from 1945 from a snake collected in Florida. By the 1970s, we have cases pretty much speckled, or I should say scattered, kind of in different parts of the eastern US. So it's been present, and overlooked, potentially predating a lot of these reports in the literature. And then based on our most recent genetic analyses of the fungal pathogen, we actually suspect that it arrived in North America sometime probably in the 1900s, up through potentially through the 2000s, just depending on the particular strain of Ophidiomyces that we're talking about. And we'll get into that a little bit more later on when we talk about origins of Ophidiomyces in North America.
So now talking a little bit more about the distribution of this disease in the world. This is a map of the current reported global distribution of Ophidiomycosis. And so all the areas that are shaded in blue are indicating both detections of Ophidiomyces fungus alone, as well as detections of fungus that were associated with clinical disease. So I haven't differentiated between those on this map, just to give you a sense of kind of everything we know so far. And this map does include both free-ranging and captive or managed snakes. So we're going to kind of start on the left and move our way across. So in North America, cases have been reported in nearly every state in the eastern, southern, and southern regions, as well as California and Idaho, and the US territories of Puerto Rico and the Virgin Islands, as well as the Canadian province of Ontario. And then moving across to Europe, cases have been reported in the United Kingdom, Portugal, Spain, France, Germany, Poland, Ukraine, the Netherlands, Austria, Hungary, Switzerland, and Italy. And then we've also seen cases in Russia, in China, and Japan, and Taiwan, and in Australia. So these have been mixes of captive and wild snakes. So specifically in Asia, there have been reports of wild snakes in China and Taiwan, and then captive snakes in Australia, Japan, and Russia. And one thing to note, so as we're learning more about these cases, they kind of pop up, but some of the captive snakes with Ophidiomycosis had been recently imported from South America and Indonesia. So those areas may have the pathogen present, but there hasn't been any surveillance reported in South America yet. So Ophidiomycosis is very likely present in more places than this map indicates, but we just need more surveillance and centralized reporting to really understand the full geographic range of the disease. In terms of snake species that we know are affected by snake fungal disease or Ophidiomycosis specifically, it has been reported in more than 60 species of snakes across 9 snake families worldwide. And you can see in this diagram on the right-hand side, I've marked the 9 snake families that have been reported with the disease. So you can see there's quite a bit of taxonomic diversity. And analyses have been done that suggest that all species are susceptible to this disease, regardless of their life history or taxonomy. So aquatic and terrestrial snakes, venomous and non-venomous snakes, it seems like everybody can get snake fungal disease. And then in addition, this disease has been reported to impact species of conservation concern. And I've listed some examples here. This is certainly not a comprehensive list. And I want to note that it does not indicate that the disease has caused population level declines in these species, merely that it has been reported in populations of these snakes. All right, back to you, Jeff.
All right, so again, we've sort of been focusing mostly on Ophidiomyces up until now, but we do want to give an appreciation for the fact that it is not the only player in these fungal infections that we see in snakes and other reptiles. So here we're looking at a phylogenetic tree of an order of fungi called Anagenales. And within this order of fungi, there's actually quite a few different genera that can cause infections in various reptile species. So you can see Ophidiomyces there in red. There's Epidemiology, which is only known to infect turtles. And then we have Paranannizzipsis and Annizzipsis, which infect a variety of different reptiles. There's Ophidiomyces is currently only characterized by a single species, Ophidiomyces ophidiicola, but Paranannizzipsis has 5 recognized or described species, and Annizzipsis has 11 described species, and those sort of vary in at least the hosts that we know of that they infect. And so we do want to talk a little bit about Paranannizzipsis because most of these reptile infecting fungi have been found primarily in captive animals, but we've now found Paranannizzipsis in wild snakes in North America. So a little bit of background, Paranannizzipsis has long been known as a pathogen in captive reptiles, both in Australia, New Zealand, and North America, known to infect lizards, tuataras, and snakes in captive situations. And we recently identified Paranannizzipsis species in wild snakes in the US and Canada. And when I say Paranannizzipsis species, it appears that there's probably at least 3 species that we found in wild snakes in North America so far. The most commonly detected of these is Paranannizzipsis australasiensis. We see it most frequently in the southeastern US. Most of the cases that we've seen have been pretty mild. The clinical signs or symptoms look very similar to ophidiomycosis. Not really able to tell them apart just based on the naked eye, but they do seem to be pretty mild to date, at least in what we've seen. However, it does seem to affect various species of snakes, and it's recently been reported to occur in chameleons that are free-ranging in Florida as well. So chameleons that are exotic but have been introduced to Florida. And one complicating factor here is that in the eastern US, when we do detect Paranannizzipsis, we often also detect Ophidiomyces on the same snakes. And so this can make it pretty difficult to get a definitive diagnosis or know which one of the pathogens is causing disease. So I mentioned that we've detected 3 species of Paranannizzipsis so far in the US. And here at this in this phylogenetic tree on the left, the strains that are bolded represent ones that we've isolated from wild snakes here in North America. So you can see again, and on the right is a map sort of showing you the distribution of these species. So Australasiensis is pretty much in the coastal plain area of the southeast so far, but we haven't done a whole lot of surveillance for Paranannizzipsis, so it's probably more widely distributed than indicated on this map. We have these 2 species that haven't been described yet then, that we're referring to just as species 1 and 2. Species 1 has only been detected once. That was in a gopher snake in Minnesota. And Paranannizzipsis species 2, we've detected in British Columbia and in Washington. And there are some conservation concerns associated with these Paranannizzipsis species. One species, which I don't have shown there, but also remains uncharacterized at this point, was one that was detected in a species of viper that has a very limited distribution, so is of conservation concern in Spain. And those infections were quite severe. Both snakes were suspected to have died as a result of being infected by that species of Paranannizzipsis. And then all the photos you're seeing on this slide are of garter snakes from the Pacific Northwest that are infected with this Paranannizzipsis species 2. And that species seems to cause very severe infections, rivaling or maybe even surpassing the most severe cases of Ophidiomyces infections that we've seen so far. And as Ellen's map showed you, to date, we haven't really detected Ophidiomyces in the Pacific Northwest in wild snakes. So it's sort of interesting that we're detecting this Paranannizzipsis there instead. But that is also infecting garter snakes, which is not a species that at least we've seen a lot of severe disease in with respect to Ophidiomyces in the eastern US. And then I just want to briefly mention here Nannizzipsis. This is not a fungus that, to my knowledge, has been detected in wild snakes in the US to date, but there have been reports, most recently in Australia, of Nanisipsis in wild lizards, causing outbreaks of very severe disease and high mortality, which was resulting in some concerns about what the impacts could be on populations of particularly eastern water dragons, which seemed most impacted. But I just think it's an example of, we're finding what we're looking for and screening for in the US. And to date, we haven't really even looked much for Nanisipsis. So it's certainly a pathogen that we want to keep our eyes open for. And then I want to sort of jump back to mostly talking about Ophidiomyces here and get into the pathogenesis, because I think understanding the pathogenesis is important, and understanding the potential impacts and a lot of other aspects, including the epidemiology and ecology of this disease. So the first thing that's important to note is that Ophidiomyces is primarily a skin pathogen, and the degree to which it causes infections, or how severe these infections are, can be quite variable. So you can see some milder cases on the left, a bull snake that just has a single lesion on the lower jaw, a water snake where there's a single scale on the side of the body that looks infected. Same gopher snake where you can see the lesion on the ventral scale there, to some more severe presentations like that eastern rat snake where the infection is involving the eye. The most classic clinical sign that we see are these necrotic areas of skin that tend to be brown in coloration. The skin's often kind of thickened and roughened, so you can see that in these photos. So anywhere from dark brown to kind of that yellow or orange brown color. In the upper right, you can see a snake that has what kind of look like nodules or bumps on the side of it. That's another common presentation that we see with snake fungal disease. And then there's a Lake Erie water snake. Sort of, you're looking at the belly of the animal in the lower right there, and you can see a pretty severe infection of the belly scales in that animal. Sorry, it seems we have a bit of a delay in the slide progression here. Okay, so these infections initiate in the outermost layer of skin. So most snakes are going to become infected from some sort of surface exposure. And on the upper right, you're looking at cross sections of snake skin viewed microscopically. So on the top there, you can sort of even see the outline of the scales. And that's a normal snake that's not infected. Below it, you can see a cross-section of skin of a snake that is infected. Again, this is on the right side of the screen. And those areas that are dark pink, kind of near the surface or edge of the skin, that's the actual area that's being invaded by the fungus. But you can also see that underneath that, the skin is becoming very thickened. The scales are sort of becoming disfigured and losing their typical shape. But it does show you how the infection is primarily limited to the skin. And then if you look at the lower, the photos in the lower part of the slide, they're demonstrating that from what we know from experimental studies, it seems that Ophidiomyces mostly seems to enter through some sort of abrasion or nick on the surface of the skin. So on the left, you're looking at a normal snake. You can see a nick in the outermost layer of skin there. And that was produced mechanically for an experiment. And on the right, then you can see how the infection is establishing underneath where that skin is compromised in an animal that was experimentally exposed to the fungus. I do want to highlight, though, that this damage to the skin is not very significant. So this is probably something that any wild snake is going to have on its skin, similar to any sort of scratch or damage that any other animal might have just in the course of living in the wild. But it does seem to provide an entryway for the fungus to start infecting the outer layer of skin. So again, these infections are sort of starting on the outside in the skin and then working their way inward. And so the main way that snakes respond or try to fight off this infection is by shedding or molting their skin. And at the bottom left there, you can see a molted skin, and the arrow's pointing to one of those lesions. That's sort of the necrotic area of skin. And when the snake molts its skin, most of the fungus is going to be cast off with that shed skin. In some cases, snakes will completely clear the infection by doing this if all the fungus is sort of limited to that outer layer of skin. But that's not always the case. So on the right, what you're looking at, again, is a cross-section microscopically of the skin of a snake that's infected. The top is the outer portion of the skin. And where that arrow is, you can see there's a white gap there between the portion of the skin that is about to be molted off and the new layer of skin that's forming. And all those little black sort of squiggles and markings that you can see in the portion of the skin above that white area is the fungus. And so when this animal molts and it's in the process of molting, you can see that most of that fungus is going to come off with the shed skin. But in this particular animal, you can also see at the tip of that arrow where the new skin is forming that there are a few pieces of the fungus that were able to penetrate down deep enough into that new layer of skin that even after this animal molts, it's going to have a new infection that gets reestablished. And it's very common that when these animals molt their skin, that they'll look completely normal again, and then potentially a few weeks later, you'll see the infection recur. So many snakes will recover after a single molting, but oftentimes we will see the infection persist, and the snake needs to molt numerous times in order to completely clear the infection. And so it becomes sort of a race between how quickly the snake can keep molting and how quickly the fungus can keep growing down further and further into the skin and beyond. And we do see the fungus go deeper into the living layers of skin where it cannot be easily shed off during a molt. And it can penetrate down beyond the skin into the muscle, into internal organs. We've even seen it go down into the bone and teeth. So it can be quite an invasive infection. But I will say that infections that progress to that extent are sort of not really the norm. They're relatively infrequent. It's sort of biased that when we get samples to the lab, often we're seeing the worst cases. And so often we do see snakes that have progressed beyond that most superficial infection. We've also seen examples of snakes that will have infections in, say, the lung, but no longer have visible signs of skin infection. So while the most common clinical signs are going to occur on the skin, we do sometimes see infections in deeper tissues where the infection on the skin may have resolved or is no longer visible. And then once the infections do invade into the deeper layers of skin or muscle where they can no longer be molted off, then the infections become chronic and the snakes can remain infected for a very long period of time. And on this slide, just showing you a few examples of some snakes that had infections that invaded deeper than that outermost layer of skin and were a little bit more severe. When snakes become infected, there's certain behavioral and physiological changes that become important to the course of the disease. So I mentioned increased frequency of shedding or molting. And snakes that are infected will molt about twice as often as a snake that's not infected. So it's quite a significant process, as you can imagine, that when snakes molt, they molt their entire skin, not just part of it. So it's a pretty big energy investment for these animals to keep up with molting frequently enough to try to clear the infection. There's also been a lot of reports of infected snakes basking or remaining out in the open more often than would be expected for a healthy snake. It's thought that these animals are probably trying to elicit a fever response. So snakes are cold-blooded. They can't generate a fever the same way mammals can, but they can go seek out warmer temperatures and try to elevate their body to induce a fever that would have the same effects as it would in a mammal, which is to stimulate certain aspects of the immune system, but also to potentially reach a temperature high enough to kill the fungal pathogen. And then as a consequence of these infections, a lot of snakes, at least in experimental trials, become anorexic and refuse to eat. And it is a common finding in severe cases that these snakes are emaciated or starving, very poor body condition. And it does make it difficult to determine what comes first. If the snake is compromised and then develops ophidiomycosis for that reason, or if the snake is infected with ophidiomycosis and then deteriorates in its health as a result of that. Probably both are true. You can imagine that as a snake's health deteriorates, if it's anorexic and not eating, it's not going to obtain the energy that it needs to continually molt, and then the infection is going to progress and get deeper, and that animal is going to sort of go into a downward spiral. There's a lot of questions about whether these infections are opportunistic or primary. From what we've seen, I would say that it can be both. We see animals that certainly have other major health issues that have ophidiomycosis, and ophidiomycosis seems, or I should say the other health issue seems unrelated to ophidiomycosis. So they're probably getting that infection secondarily. But we also see snakes that seem to not have anything else going on with them that would have ophidiomycosis as a primary cause of disease. As far as mortality, we see instances where we think the fungus is directly killing the snake. We also see cases where the skin infections allow bacteria to grow in the skin and the animals become septic and probably die of secondary bacterial infections. There's probably a high rate of predation on these animals that are sitting out in the open. So mortality is probably multifactorial when it comes to something like snake fungal disease. And then we'll get into this a little bit later, but when we're talking about pathogenesis, there's probably a difference to be expected based on the particular strain of Ophidiomyces that a snake is infected with. But again, more on that later. So in addition to just talking about Ophidiomyces, I did want to just briefly mention that we've done some experimental trials trying to better understand if some of these reptilian fungi are as limited in their host range as they appear to be, because we really only, from the literature, know of Ophidiomyces infecting snakes. So we did an experiment where we actually infected lizards with Ophidiomyces, and then we infected snakes with a species of Paranannizzipsis that's only known to infect lizards. And what we found was interesting. It turned out that lizards could be infected with Ophidiomyces and snakes could be infected with Paranannizzipsis. But you can see that the lizard that's infected with Ophidiomyces, there's no outward clinical sign that would indicate that the animal has an infection. It's only when we look microscopically that we can see that the infection is there and that that lizard is actually shedding Ophidiomyces spores. And the same was true with snakes. If we infect snakes with Paranannizzipsis, we see no clinical signs or symptoms in the snakes, but they do become infected and they are capable of potentially shedding those spores into the environment. And so the takeaway from that is that in these non-typical hosts, we might be overlooking a lot of these infections because the Paranannizzipsis does cause severe disease in lizards, but Ophidiomyces apparently does not, and vice versa. And our current surveillance is pretty biased because we're mostly sampling snakes because we think that they're the only hosts that might be harboring Ophidiomyces, but it turns out that that probably isn't true. And also suggests that we're probably overlooking some of these similar fungi that might be causing more cryptic infections but simply not really on our radar at this point, unless they were to maybe spill over into a more susceptible host. And that, of course, gets to this idea then that some of these hosts that aren't showing obvious clinical signs could still be playing an important role in transmitting these pathogens, but also in translocating them. You imagine a snake that's not showing signs if it's being moved around, either in the pet trade or even for head starting programs or things like that, not screening for these pathogens could potentially be allowing translocation without us even knowing. So back to you, Ellen.
Excuse me. So next we're going to talk about how we diagnose snake fungal disease. And we've talked about, Jeff gave a great examples of characteristic skin lesions, but we just want to point out that skin lesions can be caused by a multitude of factors. So they can be caused by trauma or other infective agents. So it's not, the presence of skin lesions is not sufficient to diagnose a snake with snake fungal disease or ophidiomycosis more specifically. And it's necessary to submit samples to a diagnostic lab for additional testing and to actually identify the pathogen that's present and causing the disease. So I have this little diagram we're going to walk through when we talk about diagnosis. We're starting out with our snake here who has some suspicious lesions. And what are our next steps? What can we do? So the first option is, not necessarily the first option, but one option, I should say, is to collect a skin biopsy. So this is a full thickness of the skin sample. And in live snakes, this should be done under the supervision of a veterinarian or another trained individual, because it should be done under anesthesia. It's a fairly invasive procedure. And you need to make sure that you're properly caring for the wound caused by taking the skin biopsy. So this skin sample can be processed through DNA extraction protocols and then used for qPCR, which is a molecular method of detecting fungal DNA. And there are currently qPCR assays for both Ophidiomyces and a Paranannizzipsis assay as well to be able to detect these fungi specifically. And just to reiterate, as Jeff mentioned, Ophidiomyces and Paranannizzipsis are sometimes both detected on the same snake. And so this can complicate diagnosing which fungus is truly causing the disease or whether maybe it's a combination. So your skin biopsy could also be used for histopathology. Jeff has shown some awesome examples of what Ophidiomycosis or snake fungal disease looks like under the microscope. These thin tissue sections that are stained and examined under the microscope. And within these tissue sections, we can detect both the changes in the tissue resulting from the infection and the host's response to the fungus, as well as the fungus itself. And then finally, we can use our skin biopsy to do a fungal culture. And this is detecting live infectious fungus that's capable of growing in culture. And so an alternative sample that we can collect, so if you aren't able to take a full skin biopsy, you want to do something a little less invasive, is to use a swab. And so the swab is essentially a sterile big Q-tip, a cotton-tipped applicator, and you rub that all over the skin of the snake, every part of the snake, the face, the belly, the back, the tail, and you want to focus on any lesions that are present because that's usually where the fungus is going to be concentrated on the animal. And so I typically make, I call it 8 passes along the snake's body. So that's like back and forth along the dorsum, the ventrum, and both sides. Snakes are tubular, but I consider them to be rectangular for these purposes and have 4 sides. And then I make sure to swab the face, of course, being careful if it's a venomous species in a tube. Sometimes you can't swab the face, and that's okay. And then I revisit any lesions that are present and kind of dig into those a little bit. And usually my swab is dirty when I'm done swabbing, and that means that I've really done a good job rubbing all over the body of that snake. So then this swab can be processed again for fungal culture, as well as you can do DNA extraction and qPCR. So I just want to note that it's important to kind of understand the interpretation of each of these tests, knowing that qPCR detects fungal DNA, but that may not be live infectious fungus. It could be DNA contamination from dead fungus that's not going to be infective. Whereas fungal culture takes a little bit longer than qPCR, but it will tell you that there's live infectious fungus present on that animal. But qPCR is kind of our go-to these days because it's typically the fastest method, and it's very sensitive and specific for these pathogens that we're looking for. So there are some other diagnostic approaches that have been investigated more recently and found to be useful when used alongside the tests that I just mentioned. UV light can be used to detect skin lesions on snakes. So if you place the snake in like a dark box and shine a UV light on it, if the animal has lesions on its skin, you'll see what the pictures on the right-hand side here, what they look like, where there's that sort of light green glow where the lesions are present. And then just to really emphasize that this is not detecting the presence of fungus, but detecting the pathologic change in the snake's tissue resulting from, it could be anything in this situation. It could be a fungal infection or it could be another cause. So once, when you use UV light, you want to then, it helps you to find the areas that can be targeted for additional sampling, either with a biopsy or with a swab. And then another option, and Jeff showed a great example of a shed skin. So we found that we can use shed skin as a diagnostic sample, both to identify the presence of skin lesions and to perform molecular testing for fungal detection. Snake sheds are nice because you can stick them in a Ziploc bag and put them in the freezer and use them for studies or for kind of retrospective analysis of, hey, my snake I have all these sheds in store, but today it has a lesion, maybe I should test some sheds and see if there's been fungus present. Because as Jeff mentioned, that fungus kind of concentrates in the shed skin as the snake tries to get rid of the fungus. And in one study, we comparing sheds to swabs, we found 100% sensitivity and 90% specificity for detection of Ophidiomyces with qPCR between these two methods. So shed skin is easier to just pick up out of an enclosure than actually holding an animal to collect a swab. So it is an option for kind of a less invasive diagnostic. Of course, being careful if you pick up sheds in the wild, and they test positive for Ophidiomyces, that may have been from a snake, or it may have been from environmental contamination as well. So just kind of a caveat there. And so because this is a relatively newly described disease, we're still figuring out ways to describe and communicate and compare our observations. One way of doing that is through these ophidiomycosis categories, which have been developed to combine observations of clinical disease and results of molecular and histopathological testing. So for example, we can more clearly communicate that some snakes have fungus detected, but no lesions, and other snakes have lesions without fungal detection, and other snakes have both lesions and fungus present. So different systems have been developed by different groups with different terminology, so I'm not going to get into the details of those today, but I just wanted to mention that lesions can be identified by visual exam or histopathology, and fungus can be detected by qPCR or fungal culture. Histopathology is great because we can confirm the presence of lesions and fungus, but you want to remember that onogenalian fungi look pretty much the same under the microscope in a tissue section, so you really want to do additional molecular testing to identify whether you're dealing with Ophidiomyces or Paranannizzipsis or a different fungal pathogen. And then the other kind of system that we're working with are these disease severity scoring systems. These have also been developed in different ways by different groups to quantitatively describe disease based on the number, size, location, and type of lesions that are present. So animals are assigned different points based on like how many lesions they have, how big they are, where they're located, and then you add that up and say, okay, this snake is a 12, that's going to be more severe than a snake that is assigned a 6, for example. And this is really useful for comparing disease severity among individuals of the same species, but it gets a little bit more complicated when we compare between species, which we know can have slightly different pathogenesis of the disease. And then to note that most of these disease severity systems only incorporate disease on the outside of the snake. So while they can be used for both live and dead animals, they don't account for internal disease. And so that's like Jeff mentioned, we see cases where the external disease has resolved, but there's still internal disease. So just something to keep in mind. Back to you, Jeff.
All right. So I alluded that we would sort of come back to looking at where we think Ophidiomyces came from, if we know now that it was introduced to North America. And there's also some other valuable things to look at here that can help us understand why snake fungal disease may be emerging and the impacts that it could have. And so on the right here is sort of a complicated figure, but you're looking at a phylogenetic tree of strains of Ophidiomyces. Most of these are from wild North American snakes. And what I want you to sort of focus on are these the areas of the tree. So you see highlighted there, or blue colored 2D. It says clonal lineage 2D, highlighting sort of a broad flat area on the tree. And if you look down lower on the tree, you see pink and also a green area where there's like no genetic diversity at all within the fungal strains. And what those are pointing out is essentially populations of Ophidiomyces that are expanding clonally. So when a pathogen is introduced to an area, usually there's only maybe one genetic strain that's introduced at a time. And in fungi, and I'm not going to get too much into the mating system of fungi, it's a little complicated. But what I mostly want you to understand is that in Ophidiomyces, like most filamentous fungi, in order for sexual reproduction to occur, you need to have complementary mating types. You need to have mating type 1 encountering mating type 2, which is shown there as MAT1-1 and MAT1-2. So these are sort of like invertebrates having male and female, right? If you want sexual reproduction to occur. So based on our genetic analyses, we believe that there were probably at least five separate introductions of Ophidiomyces into North America. And three of these are most important. And the most important of them all is this clonal lineage 2D. So I mentioned that we had that snake from 1945 museum specimen we had confirmed as being Ophidiomycosis. That snake actually had a strain from what we call clonal lineage 2E, which is those pink triangles on the tree. And we believe that lineage 2E and 2F were potentially introduced to North America first, but they're both of the same mating type, right? They're both mating type 1-1. And so they couldn't reproduce sexually. All they could do was expand clonally. And when they expand clonally, there's no genetic diversity that they really accrue, or very little when they undergo that process. Once clonal lineage 2D was introduced, that is now mating type 1-2. And that allowed sexual recombination and sexual reproduction to occur within the fungus. So on the tree, anything that's not a dot, where you can see that an isolate is bars, and the bars are different colors, those represent strains that are actually hybrids. So they represent a sexual recombination event between strain 2D or clonal lineage 2D and either 2E or 2F. So it was really that introduction of clonal lineage 2D into North America that is now allowing Ophidiomyces to evolve, because it's able to sexually reproduce and pull genes from any one of those parent strains to form new combinations of genes that allow it to survive and potentially be more virulent. And so one of our big questions looking at these data then was, when did these different clonal lineages arrive to North America? And unfortunately, we can't determine that for most of them based on the sample sizes we have. We just don't have enough isolates for anything other than clonal lineage 2D. But as I mentioned, clonal lineage 2E, we have confirmed at least in a museum specimen from much earlier. But what I want you to look at on the left here in this analysis, these are all strains from this lineage that are expanding clonally that we call 2D. And you can see the states after the different strains there, the state abbreviations. You can see that this clonal lineage 2D is very widespread in North America or Eastern North America. It looks like my labels fell off, but the dots that don't have a state label on them, those blue dots are from captive animals. So if you can look, if you can see where sort of the tree has a node or converges for the strains that have been found in wild snakes in North America, you can see that if you look down at the bottom that they share a common ancestor, we predict sometime in the 1990s. So pretty recently that this clonal lineage 2D seems to have been introduced to North America and then spread pretty rapidly throughout the eastern US. And that's important for two reasons. First, you know, 1990s is when snakes are becoming very popular in the pet trade. We're seeing a lot of importation and movement of these animals around. So you could see a mechanism by which this fungus is spreading and maybe escaping into the wild. But second, the introduction of clonal lineage 2D is happening only shortly before we're seeing the emergence of pretty severe disease related to ophidiomycosis in North America. So there might be something to, and this needs to still be confirmed through testing, but maybe clonal lineage 2D is more virulent, or it's hybrid, the ability of these strains to hybridize and pull genes from either parents. Some of these hybrids might be more virulent than the strains that have been here for a longer period of time. And so you're probably all wondering then where we think Ophidiomyces came from. And the most likely source is Eurasia. You can see on this map here, there is a European lineage of Ophidiomyces that's genetically very different from the North American lineage. And that's been found in wild snakes in Europe. But you can also see that of the strains that have been genetically characterized in Taiwan, or I should say a strain from Taiwan, a wild snake in Taiwan, most closely matches the strains that we see in wild snakes in North America. There also have been a lot of molecular or PCR-only detections of Ophidiomyces in Southeast Asia that you can see there by those asterisks. The one in Indonesia is actually snakes that were imported to Russia but were infected at the time they arrived in Russia, but had originated in Indonesia. So we can see that it's probably widespread throughout Eurasia. Most cases in Eurasia seem to be much more mild than some of the presentations we've seen in North America, which again, kind of follows this pattern of what you would expect, that the pathogen is co-evolved with hosts where it's native to, and they often don't suffer as severe of disease. And then with respect to Paranannizzipsis, we actually don't know the origin of that fungus at this time because there really hasn't been much sampling done for it as of the current time. So back to you, Ellen.
Thanks. Excuse me. So next we're going to talk a little bit more about the epidemiology of this disease. And I like to start any discussion of epidemiology with the epidemiologic triangle or triad, which I've included on this slide, which just points out to us, so disease is in the middle of this triangle, and then on each angle we have the environment, the agent, and the host. And so this emphasizes how factors related to each of these different sectors, so the environment, the agent, and the host, impact the development of disease. And this can be applied to any disease system, and it's an important reminder about how complex a disease system really is, that these factors have to really all come together in the right way, I should say, in order to for disease to occur. So we'll talk a little bit more about some of these specific factors on the next slide, but I wanted to start out talking a little bit about transmission of the fungus. And so transmission studies have mostly focused on Ophidiomyces at this point. And there's been experimental infection studies which have used direct application of the fungus to either intact or braided skin, so skin that was, some sandpaper was used to kind of take away that top layer of the keratin of the scale, or the fungus has been injected directly into the skin, so intradermally. And so this enables us to better understand the pathogenesis of disease, and as Jeff mentioned, suggests that a break in the skin is necessary in order for the fungus to invade. So in the wild, transmission is less well characterized. It's hard to sort of watch a snake become infected, but there are probably multiple ways that snakes can become infected. Horizontal transmission, so from one snake to another, likely occurs when snakes have contacted an infected snake or have been in contact with soil that contains the fungus. Multiple studies have detected the Ophidiomyces fungus in the soil, also in snake burrows where snakes brumate during the winter, and have found that this fungus is very stable in the environment. It survives over a wide range of temperatures and pH. So a snake that spends a lot of time hanging out in soil that has a lot of fungus in it, that fungus is going to opportunistically potentially invade through the skin of the snake. And so because some snakes increase their contact with each other during mating or have increased contact with soils during brumation, these activities can increase transmission. So we see, for example, more lesions in snakes that have just emerged from brumation, or snakes that have kind of been through the mating cycle. And then we also see evidence of vertical transmission. So that's transmission of fungus between a mother and an offspring. This has been documented in live-bearing species, and it may be more likely that the live-bearing or that the live young contacts the environment or contacts the mother, and that's how the fungus is transmitted, although it's possible that it could occur during while the young is maturing inside the mother. And we also see evidence of vertical transmission in egg-laying species, with one report of viable fungus being found on an eggshell and young subsequently developing disease. And then we see a lot of evidence of temporal and spatial variation in prevalence and disease severity. And so that's a very broad way of saying lots of studies doing surveillance in different ways have found different prevalence over time, or different prevalence among different sites and different severity as well. And so some specific factors that have been investigated and found to be associated with ophidiomycosis, lots of environmental and host factors, and many of these are interrelated. So there's seasonal effects that have been documented, for example, with snakes more likely to have disease in early spring when they emerge from brumation, or later in the fall. This is likely related to lower environmental temperatures, which are associated with lower host immune function. So Jeff mentioned that snakes are ectotherms, so their body temperature depends on the environmental temperature. So when it's colder outside, their metabolism is lower and their immune function is a little bit lower. Periods of higher rainfall have also been associated with increased disease. And the soil microbiome is thought to play a role with when certain microbes are present in the soil, there's less Ophidiomyces fungus present. And so snake species, moving over to kind of more specific host factors, snake species has been studied in relation to disease susceptibility, with higher prevalence of disease reported in Nerodia species, which are water snakes, as well as rattlesnakes. And there's a number of hypotheses regarding this. It could be attributed to the skin structure or thickness or microbiome of the skin of these individuals, as well as factors related to their genetics and life history. For example, studies have reported higher prevalence or more severe disease in aquatic snakes, which spend more time in water. And as anyone who's taken a long shower will know, you spend a lot of time in water, your skin gets a little soft, and that may, so spending a lot of time in water may compromise their skin integrity as a barrier to fungal invasion. That's just a personal hypothesis of mine to explain observations of more disease in aquatic species. Host body condition also varies seasonally with food availability, as does the hypothalamic pituitary adrenal axis, which mediates the host stress response. And then we know that snake immune function depends on sex, age, species, environmental temperature, and season. So that's known to be associated with ophidiomycosis as well. In terms of the conservation implications of this disease, this is really an area of a lot of debate and sort of a lot of more questions than answers, because the population level impacts are unclear in many situations. And they depend on the species and population in question and really require additional study. So while mass mortality events in snakes are typically not reported in relation to snake fungal disease, this is just one of many threats to snake health. Infection is just another source of stress in populations that are already facing many threats, climate change, habitat fragmentation, contamination, to name a few. And so this is an added concern for threatened and endangered species, as a possibility that it could increase the rate of decline or cause additional mortality in these species. Whether or not ophidiomycosis is an emerging disease is also a topic of debate. There's two hypotheses that explain an emerging disease. One is the emerging pathogen. One is the novel pathogen hypothesis, which describes that a pathogen is emerging because it's spread into a new geographic area. And then there's the endemic pathogen hypothesis, which explains emergence through a recent increase in host strains or pathogenicity. And so we have evidence that supports each of these hypotheses in different situations, but also fails to support these hypotheses in different situations. So we can likely say that ophidiomycosis is emerging in some areas, but is endemic in other areas. So overall, we really do need more data to fully understand the conservation implications and population level impacts, specifically increased surveillance, standardized sampling and data reporting, really doing things the same way over and over again so that we can better understand things at a larger scale. Okay, so now I'm going to move into some treatment and management options. This is also an area that needs a great deal more research, but we kind of have to think about this in terms of what are our goals for treating or managing this disease based on whether we're dealing with a large free-ranging population or we're dealing with a single individual in a managed or captive situation. So in general, it's difficult to treat fungal diseases in reptiles because of the long duration of treatment needed to see clinical improvement. So fungal diseases in general take a long time to treat, and reptiles take a long time to treat because of their lower metabolism. So you combine those two and you're really going to need to treat an animal for likely weeks to months or longer in order to see clinical improvement. So several antifungal drugs have been found to be effective against Ophidiomyces fungus in vitro. And a lot of treatment attempts have been reported with combinations of different drugs, different antifungal drugs, topical treatments, systemic treatments. Sometimes there is some success in these reports, but often the animal dies or just doesn't get any better over time. And at this point, only one controlled clinical trial has been conducted. So this trial used a drug called terbinafine and administered that drug through nebulization. And I've kind of shown what that looks like in this picture here. So we place our snake in a sealed glass aquarium and we pump in a solution of the antifungal drug. And so the animal receives the drug through direct skin deposition because it's sitting in the mist, as well as through it's inhaling the drug as it inhales the mist, and that results in systemic absorption. And so there's been some promising results with this terbinafine nebulization, as well as subcutaneous implants of terbinafine, although the implants have not been subjected to a clinical trial. So briefly, I just want to talk about the clinical trial that I conducted as part of my PhD research. So again, we used wild-caught Lake Erie water snakes with natural ophidiomycosis and separated them into groups based on disease severity score, demographic characteristics, and then progression of disease during an acclimation period. And then snakes either received saline as the control treatment or a terbinafine nebulization as the treatment. And so we saw differences actually across the 2 years of the study. So the second year of the study, we had added a second control group, which was a no treatment control group, thinking that saline nebulization could have an impact on the clinical resolution. And so in the first year of the study, we saw no animals that were treated with saline have resolution of disease. So that's the clinical signs, the lesions resolving over time. But in the group that was treated with terbinafine nebulization, 23% of those snakes had resolution of disease. And a note that those snakes were treated for several months, up to 5 or 6 months of daily nebulization in order to reach that point. And then in the second year of the study, we saw some resolution of disease in our no treatment control group and our saline control group, but a higher percentage of snakes had resolution of disease that were treated with the terbinafine nebulization. So while these results indicate that this is not a silver bullet, but it does help some snakes sometimes. And because of the long duration of treatment, this is going to be a treatment that's more useful in snakes that are in a managed population that can be kept in captivity and treated daily for months on end. But this is going to be less useful in wild snakes because you're not going to catch your snake every day or keep the snake in captivity for 6 months. And in some places, regulations prohibit release of snakes after that period of captivity. So when we think about management approaches in wild snakes, our aims are typically to decrease the environmental load of the fungus. So that's we want to decrease how much fungus the snake is encountering. So we can decrease the amount of fungus in the soil, decrease how much fungus the snakes are exposed to, or we can decrease snakes' susceptibility to becoming infected. How we do that is still an area of further study for sure. And environmental management strategies in terms of fungal pathogens that live in the environment are challenging because if you treat an environment with an antifungal substance, it's going to disrupt the microbial community and have ecosystem level impacts. Some of these environmental management strategies have been studied for Ophidiomyces and white nose syndrome. For example, they've looked at changing the environmental salinity for aquatic species or applying antifungal compounds to caves for bats with white nose syndrome. And there are some promising results of these studies, but so far no one's been trying to manage snake fungal disease at the landscape level. So what can we do? Biosecurity is so important with any wildlife disease, any infectious disease at all. And so that's kind of our best hope right now is to really just focus on having good biosecurity in the field. And when we, in captive snake populations as well. So important to remember to wear gloves when handling snakes, to clean boots and equipment between sites, and to disinfect natural materials if they're being used in snake enclosures. And then snakes that are being moved between locations or between captive populations, quarantining and testing those snakes for Ophidiomyces, for Paranannizzipsis, for other pathogens as well, to ensure that the snakes are healthy and will not transmit pathogens to their new population or collection. There has been some research into decontamination strategies specifically for Ophidiomyces. Found that effective disinfection can be achieved through 3 and 10% bleach solutions with contact times between 2 and 10 minutes, as well as a 70% ethanol solution with a 2-minute contact time. And then some other commercial products like Rocal, Lysol, CLR, Process NPD, and 409 were also effective with a contact time of 10 minutes and were successful in killing the fungal spores. Some other disinfectants that were studied but were found to not be effective were chlorhexidine, simple green, and spectracide. So don't use those and expect them to kill your fungal spores if you're going to decontaminate an area. And then, of course, with any decontamination protocol, use of any disinfectant, organic matter needs to be removed from the equipment before disinfection so that the disinfectant will appropriately contact the equipment and effectively decontaminate the surface. So this is things like mud, dirt, blood, tissue, things like that. So kind of to sum up, what are some good field practices when we're out doing field work or just hiking or exploring natural areas? Always important, if you don't have to disturb a snake, don't bother the snake. Don't touch it or disturb it because you don't want to, A, stress out the animal, and B, you don't want to potentially pick up any pathogens that that snake may be carrying. Kind of on the same line, wearing gloves when handling snakes for field work and changing gloves between snakes. Again, will reduce the amount of fungus that's being spread between animals. Housing snakes in temporary storage bags and then cleaning those, so housing them individually in bags. Don't house your snakes together when you're doing field work. And cleaning the storage bags or storage containers with hot water and bleach if you're able after each use, so between every snake. And then again, cleaning and disinfecting boots, handling equipment such as hooks, buckets, tape measures, vehicle tires. A lot of people forget about. Bleach your truck tires before you move to the next field site so that you're not moving fungus or other pathogens to a new site. What else can we do as members of the public? In addition to cleaning our gear, if you see a sick animal, there's actually what we call the herp disease alert system, which you can reach out to and report situations where you see dead or sick looking reptiles and amphibians. This is the email address that you can reach out to. You can use the QR code that's on this image here. And that will then email a group of researchers who then can contact your local, state, federal authorities who then can investigate that mortality or morbidity situation and figure out what's going on. Back to you, Jeff.
All right. So just to kind of wrap up here, we wanted to talk about some of the major knowledge gaps and where we think research is going with respect to snake fungal disease. And I think the first big thing here, and Ellen mentioned this when we're talking about conservation implications, is better understanding what the impacts are on populations. Because we lack this historical data on snakes, we don't really know what population trends look like. And the studies that have been done trying to understand impacts of Ophidiomyces are often short term. And as I mentioned, that this disease can be quite chronic. So things like chytrid and white nose syndrome have sort of shaped how we view fungal diseases and thinking that they act pretty acutely, that you can have large numbers of animals die in a short period of time. Population declines happen very quickly. But with something chronic like ophidiomycosis, that might not be the case. And so we might need more than just a year or two of data to really understand what's going on in a population. There's also variability likely, at the geographic level, at the host species level. So trying to extrapolate what happens in one population or one species in one area really becomes difficult. And then throwing on top of that, this idea that there's different strains of Ophidiomyces, and if some are more virulent than others, we could be monitoring a population that doesn't have a very virulent strain, and trying to extrapolate what's happening from there really doesn't make much sense. So I think there's going to have to be a lot more work done on trying to understand what the actual population impacts are and where those impacts are likely to happen. Also, this concept of how Ophidiomyces is evolving now that it's able to reproduce sexually in North America, and how that's going to affect disease severity and outcome, whether that could actually be the mechanism by which we're seeing more severe disease in some instances or in some populations. Also, if it could be evolving in ways that might allow the pathogen to expand its distribution, the species of snakes or species of reptiles it can infect and the severity it causes. When Ellen showed you that distribution map, you could see that there are parts of the US where we haven't really detected Ophidiomyces yet. And maybe it's just because the strains we have here aren't very well adapted to conditions in those areas, but that could potentially change now that the pathogen is a little bit more free to evolve. And again, as Ellen touched on, understanding what effective management strategies are. Eradication is probably impossible for snake fungal disease. And so what type of intervention can we take that maybe reduces impacts on snake populations and maybe gives them the tools that they need to recover from infection and that sort of thing? Trying to rally public support for snakes. I'm sure everyone here realizes that snakes don't really carry as much public favor as some other wildlife species, but also making the public more aware of how their actions might be involved in spreading these diseases and things like that. As Ellen mentioned, sharing information, I think, becomes important between states or different management agencies. That if you're seeing effects, letting other managers know so that they can be on the lookout and sharing information that way. And then finally, getting at this question of how, maybe being more proactive instead of reactive. How are things being moved around? How can we prevent these translocations from happening? How can we prevent new strains that might be more devastating from reaching North America? And also looking at not just these fungi, but reptile pathogens in general. What are we missing? There's probably a lot of things out there that aren't causing disease in their hosts in their native countries or their native locations, but as we move things around, those pathogens are getting moved around as well and have the potential of having pretty devastating impacts if they get introduced to new areas. So we are going to end it there. Apologize for running over. Even in going long, I think we probably still have just scratched the surface and there's probably a lot of questions. So we are happy to stick around as long as we can to answer any of those.
Thank you so much, Jeff and Ellen. That was awesome. We have been fielding a lot of questions in the chat. So if any of your questions were answered, we are not going to repeat them here now just for time's sake, but we will add them to a list that will be available along with the YouTube video. Also, if your question is not answered because of timing or you're watching the recording, we, the PARC Disease Task Team, will be gathering additional questions into a single document to send to Jeff and Ellen, and we'll include our email in the chat now, as well as the caption for the video. So I think the best way to move forward is you are able to unmute yourself now. So I think we should field questions from the live audience, and then we can go to the chat real quick for the ones that weren't answered. And if no one speaks up, we can go right to the chat one. All right. Okay, so I'm going to go to the chat questions that weren't answered. One of them had a lot of discussion. It was about biopsies and transmitter use and how that can impact or increase SFT infection. So the question is, if taking a biopsy, are there concerns for replicating the type of damage that we see in experimental infections, potentially leaving wild snakes vulnerable to become infected? And are there any recommendations if that is the sampling approach? And such as limiting the sample collection for extreme cases where release wouldn't happen. And I know that Corinna talked a little bit about transmitter implantation and how that can kind of cause the disease to proliferate. But Ellen and Jeff, did you have anything you wanted to add to those questions?
Sorry, I may have lost track of the multitude of questions. I think the question was about transmitters and whether transmitters should be used in experimental studies because of that was not the question. I'm sorry.
No, you're totally fine. It was a few questions, so I should just kind of streamline it. It was just talking a little bit about, if you're doing sampling that is going to damage the snake, what are the recommendations for the sampling approaches to kind of limit further infection? And Corinna, correct me if I'm wrong.
Just kind of go over the risks of like biopsying a snake with active lesions.
Yeah, so typically that's not going to be recommended, right? Like if you are in the field and you have a snake that's severely impacted, maybe do a swab instead of a biopsy. Biopsies are more useful for getting that tissue sample for histopathology to look at it under the microscope and see the fungus and see the changes. But I would not recommend that in a severely infected animal who could not be then treated individually following the collection of that biopsy. So if you had for example, a snake in a managed collection that you could treat with topical antifungals or antibiotics, then I would be more comfortable taking a biopsy in that situation.
Yeah, I would agree with that, and I would say that you know, I think for field sampling, depending on the reason you want to collect the biopsy, a lot of people now, if for example they want to get a culture of Ophidiomyces from that sample, they'll actually just remove part of the necrotic skin and not cause any sort of new open wound, or they'll do a swab of the lesion, as Ellen mentioned. So there are alternatives. I don't know that the biopsy is as useful anymore as it may have been early in the investigation when we were trying to look at what was going on in the skin in general, especially if it's in an uncontrolled environment where you can't monitor or keep the snake or easily, disinfect it.
Awesome, thank you. Another question we had is, are juvenile snakes more susceptible to snake fungal disease or more likely to die from an infection?
That's a good question. My only experience I can offer there is saying that we've seen juvenile snakes die very quickly. Like we've seen snakes that are born and then within less than a week are dead with really severe lesions, but I don't know the mechanism behind that. I don't know what Ellen's experience has been.
Yeah, I would say, I mean, certainly reasons for juvenile mortality are numerous, whether that's, the just failure to thrive and failure to succeed as an individual, whether that individual, I mean, certainly juveniles are going to have less immune function than an adult. So that may contribute to mortality or to increased disease severity. I don't specifically have data to support that they're more susceptible or more likely to die, but it's a great question.
Can I ask a live question?
Yeah, of course.
All right, so I'm playing devil's advocate here. So let's imagine instead of talking about snake fungal disease like it's chytrid fungus or white nose, let's imagine it's athlete's foot, something that we don't do much about or worry about in most of the population, but could be an issue in certain populations and could possibly kill something if we expose them in a lab in the right conditions. And so if we were worried about athlete's foot, I don't think we'd be decontaminating ourselves every time we went to be anywhere it could be. And so given the maps of where it is, I think we all have this mindset of alarmism that this is really bad. And you had mentioned hope at some point. I just want to be the devil's advocate and say, why should we be alarmed when we've seen 2 potential population declines and we see the source? And I mean, you didn't present any numbers, Jeff, about how many snakes you're getting in. And you said that most cases aren't really bad. I mean, can someone address this? And do you have enough proof to say this is really bad and we should be worried about it instead of it just being something that's here?
So, I mean, I have a few thoughts on that. One is that we certainly see cases where, this is certainly like much more of a severe disease than something like athlete's foot. I think that it's very difficult to study because I think that outbreaks are somewhat inconsistent. There's probably a lot of environmental factors built in. The idea of whether, the concept of it's there and what's the likelihood of moving it? Like, is there a concern of moving it to a place where it already has been? That's where my concern for new strains come in, but I think it extends beyond just these pathogens. And I think when we're talking about, handling wildlife for research projects in general, it applies to what we don't know that animals might be carrying and what gets moved around before we even necessarily know that it exists. So the things that Ellen mentions are, I would say, more or less really just best practices in general for working with wildlife, trying to limit transmission of pathogens. But as far as data, hard data on population declines go, the answer is no. But I would also argue that I haven't seen good studies that have followed snakes that suggest that there aren't potentially some impacts that are happening there as well. So I think it's a question mark, and it comes back to like the better safe than sorry, not just for this pathogen, but for any pathogen for that matter.
Yeah, I agree with that. I think while in some populations you could equate it to a mild fungal infection like athlete's foot, where we do see snakes, they get sick, they shed, they look better, doesn't seem to slow them down. There are certainly like reported populations that have suffered extremely severe disease in association with population declines, whether that was the cause of the population decline or not. Eastern Massasauga rattlesnakes in Illinois have shown extremely severe disease and have had population declines to, the original kind of timber rattlesnake study in New Hampshire. So I do agree with Jeff that this is, a more severe situation than I would call athlete's foot, which, just kind of bugs you for a little while and it goes away. But I don't necessarily think that we need to be treating every individual snake. I mean, disease is a natural aspect of an ecosystem that waxes and wanes over time. And animals, over time will develop coexistence with pathogens. So I'm certainly not a proponent for pulling in every diseased snake and treating it for 6 months, just to make sure that its lesions go away. But I do think, we need to keep in mind the multitude of threats to snake health and population health, and bear in mind that, this is one more thing falling on snakes that, in addition to climate change and habitat fragmentation and all those other factors that are impacting, population health in snakes. So I do think we need more data. It's inevitable that pathogens are going to get spread around. That's not, certainly, telling people to bleach their truck tires is not going to prevent this from getting spread. It's going to spread in one way or another. But I think making people conscious of how their potential for moving pathogens around into new systems is important, not only for Ophidiomyces, but for more sort of those crisis fungus like white nose syndrome or chytrid or ranavirus that can have really severe acute widespread effects on wildlife populations. So it's a little bit bigger than this, is why I kind of stress all that biosecurity, but it's also the veterinarian in me that wants to make sure we're not making anybody sick that doesn't need to be.
I just bring it up just because there's a lot of wildlife people in the room, and it does actually become fairly burdensome to bleach all of your supplies and your field gear. And I have not seen enough proof that would make me take that much of an issue with it. I think a lot of it is alarmist. And it does affect field work, and it does take time and resources that make it off-putting. And the correct answer is somewhere in the middle. I understand a lot of the things you're saying about lab safety, but sometimes I feel like we're talking about lab infections and then making assumptions in the field based on these exposures that are occurring in really weird environments and in very extreme amounts of the pathogen. I don't know. I think with a lot of graduate students and recent graduate students and undergrads on here, we do need to think about both sides of the argument and think realistic about what this disease is.
We appreciate that perspective, John. Thanks.
Yeah, and I also think, like Ellen and Jeff were saying, there's a huge gap in knowledge of progression of disease. And so we don't really see how these chronic and maybe sublethal infections are impacting populations long term. And that's what we really need to kind of understand instead of just tracking just where it is. So there's a lot more research to be done to understand the threat. Another question that we
Hear,
had.
hear.
Thank you. Yeah. So another question that we had, just going to ask a couple more so we don't stay on too far past 5. I thought this one was interesting. How would you interpret if a shed was positive, but swabs were negative? And she kind of had more context of, did the snake clear the infection or did the shed get contaminated by the environment? So what would, Jeff and Ellen, what would your interpretation be if you had a shed that was positive, but the swabs were negative?
My inclination would be that we talked about the fungus being concentrated in that shed skin. And so I think that the test may not have been sensitive enough to detect what was on the swab, or there may have been so little fungus actually left on the new skin to detect with a swab in qPCR. So I think that's just kind of, we're talking kind of a sensitivity issue here. Or, you know, that the fungus that's still present on the animal may be, you know, in deeper tissues. It's no longer on the surface of the skin, so you're not going to pick it up with the swab. You know, would I treat that animal if it still had lesions? Maybe, but if they didn't have lesions, I probably would just monitor that animal if a swab was negative. But that's a kind of a judgment call depending on the situation and what's been, you know, if the snake had really severe lesions and then it shed and the shed was positive and the skin was negative, probably would keep treating that animal. But it's, you know, happy to talk about that more in detail. It's definitely kind of a case specific situation.
Yeah, thanks. Sorry, I just wanted to jump in because this was my question. I was coming at it from a perspective of like a veterinarian in a zoo situation where, you know, you find a shed, it looks a little funky, you test it, you don't even know which individual it came off of.
Okay.
You find something in it, right? This isn't something that's happened to me. I'm sort of thinking from a management scenario of then what do I, and we can talk about this offline, sorry. I know we're running low on time, but it just
Yeah,
goes
feel free to reach out to me. We can talk for days.
Question to me, right? Like, what do you tell your keepers to do? Woohoo! So thank you for addressing that.
Yeah, for sure. Feel free to reach out, Ellen, and we can chat more.
I do think it's worth pointing out there, though, that, you know, a negative PCR result never truly means that it's not there. And I think in snake fungal disease, even more so than in some other fungal pathogens like chytrid and white nose syndrome, that these fungi aren't always like immediately at the surface and able to be picked up by the swab, sort of depending on the stage of the infection. And so, you know, you might want to swab asymptomatic snakes or even symptomatic snakes several times before you conclude that the organism isn't there.
Yeah, thank you. Yeah, for sure. I you know, I think that's the reality that we're always up against as clinicians. Just sort of when you don't have the positive, but also knowing in the context that negative is never really negative, trying to formulate a plan that is feasible and also acceptable for, you know, your staff sometimes becomes a fun scenario.
I don't know how many more questions you want to take, Jeff and Ellen. Maybe we go till 5:10, 5:15, just cut it off at 5:15 the latest. But there were some questions in the chat about hydrogen peroxide, and we were kind of chatting about this prior to, but they were kind of curious if higher concentrations of hydrogen peroxide would kill the fungus on tools, substrate, and other such tools.
I will say that I don't have any specific data on that. That wasn't one of the disinfectants that was included in the Redkowska's study. So I would be hesitant to tell you that it probably does something, but I wouldn't rely on it based on the data that we have.
Yeah, I mean, I think there's a lot of substances that you can, kind of make some assumptions about, but it's hard to like recommend something when you don't, know for sure if it works or not, or it hasn't been tested.
Another question that we had, and I don't know if we will have a specific answer to this, but there was a question about if the SFD strains in Idaho were different than what has been found in the eastern United States. There was some concern and confusion with people in Idaho if they have a distinct strain. I don't know if you know that off the top of your head.
So we don't have any samples from Idaho that have been positive. I think we had one, and this is another thing. There are other fungi out there that seem to cause disease. The only snake I know we've got from Idaho did have lesions, but it ended up being a fungus that was not Ophidiomyces or Paranannizzipsis. I don't know if Ellen has processed samples from Idaho.
Yeah, the positive reports from Idaho were from a military installation, and so that was a positive detection of Ophidiomyces from a swab. I don't recall off the top of my head if the animal or animals had lesions as well, but it would have been, to report a positive in that way, it definitely would have been positive on qPCR for Ophidiomyces. So, but unfortunately, that sample was not included in the genotyping work. It's a great question. I don't have a specific answer to that.
Excuse me. May I ask one?
Yes, go for it.
Hi. Yeah, this is Tigran Tadevosyan from Ogilvy Associates from Massachusetts. Thank you for great presentation to all of you guys for organizing this, Jeff and Alan. So my question is, I actually posted it over there, and I got a little bit impatient. Yes. Is it possible that, you've mentioned that in some species, we don't really detect higher prevalence, like, for example, in some garter snakes. Is it possible that our low detection of prevalence may be linked to low survival rate and, just faster duration of a disease in some species as opposed to others, especially in the smaller ones? What is your perspective on that? That's the question. Thanks.
I don't know the answer to that. I mean, obviously, host susceptibility is caused by, can have a lot of underpinnings to it. One of my, I guess one thing I've noticed is that, and there are some exceptions, a lot of the smaller bodied snake species, maybe even ones that tend not to have as long of lifespans, are the ones that we have not seen as many severe cases in. It seems to be kind of like the larger bodied, longer lived species. Ellen mentioned pit vipers. We see it in a lot of like the rat snakes and things like that. So one thought could be if some of these smaller species maybe, molt more frequently, maybe there's something to do there. I don't know though. I mean, I couldn't say for sure, like, what's controlling that or what's affecting that. And I also don't know that we have good broad geographic coverage, across species and areas to really say for sure that some species have lower prevalence. It's just kind of an observation based on what we get submitted. But, I think that was a question when someone brought up young animals, and, it's a good question, like how many young animals might disappear off the landscape before you even, see them actually being infected. So.
Yeah, actually, I've been observing it for a while, and my impression was that, some species just live longer with it. And we consequently see it more frequent in those species, while others we just don't know because we only see sort of what we see and not severe cases. I guess. Thank you, Jeff.
Yep.
I would chime in and say that, like Jeff said, prevalence estimates are tricky because they depend on so many different factors. They depend on sampling techniques, seasonality, species, geographic location, factors about the soil. And so when we say, one area had a higher prevalence than another, there's so much about the sampling protocol that can impact that. So I think it's interesting if we can say, okay, we were in this area at the 4 different times and we did see a higher prevalence in this species consistently compared to another species. But, while, if low prevalence were due to causing more mortality, I would expect that we would see more dead animals, going out and being in the field, although sometimes, dead snakes are picked up and predated or just disintegrate pretty quickly. But yeah, it's an interesting thought for sure that we're seeing lower prevalence because those animals are dying, and it's somehow more acute and more causing more mortality in those species than the more chronic situations.
All right, we're gonna cut the questions off there so our speakers can get some dinner. Thank you, Ellen and Jeff, so much. That was a wonderful webinar. Thank you all for attending. We will stop the recording now, and we will post this on our webpage and our YouTube.